They are unicellular organisms. Like I said, it's like they are living in one room apartment. Inside that room, everything has to be there. Think about one room that has no living room. It's just one room. So everything has to be there. So that means they don't have compartments. They are not divided into compartments. Everything is just there. So compartment is where you have endoplasmic reticulum, the organelles separate in different membranes. But prokaryotic cells don't have that luxury. So everything is inside the cytoplasm. And then they have a region at the middle of it where the DNA will be located. So for prokaryotic organism, we don't say they have nucleus. Eukaryotic have nucleus, but prokaryotic cells don't have nucleus because they don't have membranes that covers them. They are small, they are tiny, they are less complex, so they are simple. And their DNA is usually circular and it's just one, even though it's long and twisted on each other, but it's one. Now, so they are unicellular organisms, they lack nucleus or nuclei. They are much smaller, like I said. You see them everywhere. There is sufficient moisture. And in theory, this is one of the reasons that it's difficult for bacteria to grow on your skin because our skin does not host moist for them to be able to survive in because of the keratin that is on the skin. All right. Now, so they reproduce asexually. Bacterial cell walls, they contain peptidoglycan, which is one of the reasons I will be talking about this later, which is one of the ways that we divide bacteria into, classify bacteria into gram positive or gram negative. The gram positive, the peptidoglycan layers determines which one will be positive or which one will be negative. I'll talk more about that later. Now, archaea cell walls are composed of polymers other than peptidoglycan. So they don't have peptidoglycan in their own cell wall. They have, unlike bacteria. All right, so let's go. Now for fungi are eukaryotic. Unlike bacteria and archaea, they're prokaryotic. I told you other ones are eukaryotic organisms. So they have a membrane-bound nucleus, which is what differentiates eukaryotic organism. Again, prokaryotic organisms do not have true nucleus. They only have what we call nucleoid region. All right, they only have what we call nucleoid region. Whereas eukaryotic organism, which is where fungi belongs to, they have nucleus, and the nucleus has the membrane. Now, they don't make food. They obtain food from other organisms. So usually, we classify them as decomposers. We'll talk about that more later if you have to. So they have cell walls. Like plants, they have cell wall. They include modes, which are multicellular, and they also have yeast that are unicellular. Now, the modes, they are able to reproduce sexually and asexually, and the yeast only reproduce using spores and bud as well. Now, protozoans. Protozoans, they are also single cells, and they are eukaryotic organisms, not prokaryotic. Now, of course, you know that there are some organisms that are unicellular, eukaryotic, and there are some organisms, prokaryotic cells are usually one cell, but there are eukaryotic cells that are one cell, but they are mostly multicellular. So protozoa is eukaryotic, meaning that it's not as simple as bacteria and archaea, but it is a one-cell organism. They have the appearance, they look close to what you see in animals, and you see them in water environment more. Some of them, they have animal hosts, so they live on their host. They reproduce sexually and asexually as well. Now, some of them here, you see them as locomotion means movement. So most of them are capable of movement. They are able to move. They use sorry, I'm trying to close this on my computer. All right. Now, if you look at the first image here, the image A, they use pseudopods to move. All right. So that's pseudopod. They use pseudopods to move, and that's their nucleus there. Now, these guys move with cilia, and these ones move with flagellum. You can see the flagellum there, right there. So those are protozoans. Then algae could be unicellular. They can be multicellular as well. And of course, they are eukaryotic organisms. They are photosynthetic. So you have different algae. You have green algae, red algae. They are photosynthetic. They are not as complex, but some of them exist as unicellular. Some of them exist as multicellular organisms. Okay. Now, there are generally we have some of the microorganisms. We call them microbes. So when you hear microbes, microorganism is the same. Now, we'll be talking about them over time, but some of them, they are parasites. Some of them, we regard them as viruses. So sometimes we just regard some, you just see them called parasite, like this worm now, we call it parasitic worm. Then we have the virus as well. Now, this is a virus, and of course, you know that the key thing about virus is that they get into the host and then they manipulate the DNA of the host so that they replicate themselves. So in quotes, they use the resources of the host to replicate themselves. And that's one of the reasons that viruses are difficult to kill, because anything, what they do, they get into the host and mimic the host. And so the host is funding them in quotes unknowingly, because they get into the host and mimic the cells of the host, the DNA of the host. So they look like the DNA, and this host begin to spend on them, make them grow at the detriment of itself. So that's what viruses does. And so it's difficult to kill them because any medication you take to kill them will kill the host cells as well, because they already mimic the host cell. And so I used to crack this joke, like it's more like a joke. During when COVID came forth, of course, COVID is a virus. So when COVID came out, a woman from my country got online and she said, "Oh, now I have a solution to this bacteria." And so immediately I heard that, I zoned off, because the moment you are calling virus a bacteria, it means you don't even know what you are doing, because it doesn't take anything to kill a bacteria. Bacteria kills with antibiotics. You kill a bacteria with antibiotics. It's a virus that is difficult to kill, and that's why you notice that all the viral infections, what we have for it are vaccines. It's not medication. It's vaccines, because any medication you take that attack the viral cells, the viral DNA will also attack the cell in the host, because they mimic the host. All right. So, golden age microbiology. Like I said, I'm just doing quick run through. Wherever we stop there, that's where we stop for the first chapter, is just to do like a little introduction to the class. Now, scientists search for answers to these 4 questions, which are the things that actually characterize the present day microbiology. So the question is this, which of course this is also biology: is the spontaneous generation of microbial life possible? Now, the thought that microorganisms just evolve spontaneously without any precursor, is that possible? Of course, biology says cells exist from pre-existing cells. Now, is spontaneity something that is believed to happen with microbiology? Is it possible that you cover something perfectly well and then microorganism will start to grow in it spontaneously without being exposed? All right. Now they also want to look at it. What causes fermentation? How does things ferment? Now, what also causes disease? How can we prevent infection and disease? So those are the things that researchers has gone into and brought some answers to. So what I would do is I will quickly look at few of them in a bit, and I will stay on some that I feel we should stay on before we close. All right. Now, so does microorganism evolve spontaneously? Do they just evolve from nowhere? Now, some philosophers, scientists in the past, they have been with the impression that microorganism came from asexual reproduction, which is what we're looking at from those classification of bacteria, archaea, protists, protozoa, fungi, that some of them do asexual reproduction. Some of them do sexual reproduction. Then the third one, which is the questionable one, is non-living thing. So they are saying that microorganisms could arise from asexual reproduction. It's logical to us as scientists. They're saying that they could come from sexual reproduction, okay, but that they could also arise from non-living matter, which is where we want to look at. Now, so Aristotle was the guy that started in spontaneous generation thing. He's the one that says uh, microorganism can exist, can just come off from non-living matter. Now, look at what Reddy's experiment taught us. So this guy want to, so it's more like they have Aristotle's belief and they want to criticize that, so to say. So everybody started having their research to see what they can use to defend or to contradict what Aristotle had said. So he did his experiment and then he said when they came, it was kept isolated flies. So you see, on this here, he had meat here, and the flask he used was opened, whereas here the flask was sealed. Here he closed the flask with just like a fiber cloth. Now, by the time he did that, he he saw that the one that was covered, that maggot did not grow there. He saw the one that was exposed, maggot grew there. Now, so because this one that was totally closed, maggot did not grow there, then they were now beginning to doubt that, okay, so if there is spontaneous um if the issue of spontaneous origination actually happens, then they should grow here. So they are beginning to now think that when there is no exposure, there was no growth of microorganism at all. The only time when there was growth is because there was direct or indirect exposure, which is what happened in this third one and in the first one. So that was the beginning of loopholes to Aristotle's view. All right. So the next one was what Needham also did. So he also wanted to follow up on that to see if this actually happened. So the first thing he did is he used like beef gravy as well. And then what he did is he briefly boiled some of the broth and then he left the container open. So now, when the fact that he briefly boiled it means that the bacteria may not have died. Anything that is there may not have died very well because it was not a deep boiling. It was not a deep cook. It just briefly boiled it. So he sealed the flask, and then he noticed that bacteria grew in it. And the thought is that because he briefly boiled it, he may not have. Let me see if I can use my writing pad here. I got one last semester that I'm still going to get used to. What is this? Please give me a moment. Oops. Just a moment. Okay. Sorry. I'll leave it. I'm not getting it now. I'll check it for that time. So the key thing was that he briefly boiled it, and the fact that he briefly boiled it, that implies that the bacteria may not have died. That's the key thing here. So the thought is that the microorganisms grew on here all because they were still there, because they were not fully dead. All right, that was the issue here. Now, so does this support spontaneous generation? Microorganisms flourish in various soup that has been exposed to the air. It claimed a life source was present in all inorganic non-living matters. Let's go. I'll talk about that together on the next one. So somebody else, Spallanzani, then went ahead and did another experiment. So he did different variations of what this guy did. Needham only did one, right? So he did 3 batches. Now the first one, he boiled the soup for 1 hour, and then he covered it. Having boiled it for 1 hour and covered it, the soup stayed sterile. There was no microorganism that grew up there. Everything stayed intact and pure. Second batch, he boiled the soup for just few minutes. He realized that microorganism grew there after some days. Then the third batch, he boiled it for 1 hour, but he didn't close it properly. So he left it open a little bit such that it can be exposed. Microorganism also grew there. So in essence, what he came up with to fault Aristotle and also to further a little bit of work on Eden was that the soup that was totally boiled very well, the bacteria in it or every other microorganism in it was killed with the heat. And then they were covered fully such that no microorganism could enter into it. And then they saw that no microorganism actually grew in it. But the second batch that was boiled for a few minutes, the thought is that the microorganism did not die, and so they could grow. And then the third batch that was also boiled for 1 hour, just like the first batch, because it was left opened, microorganism grew in it because they had access to come into it. All right. So it concluded that Needham, which was the guy that boiled the meat briefly, failed to heat vias sufficiently to kill all the microbes and had not sealed them tightly enough. That was what this guy concluded on. Now, he then said that microorganisms exist in air and can contaminate experiments. And that was why he covered the first one very well. And the third one that he didn't cover very well, the microorganism that exists in his thoughts, got access into that. And then that was why they grew up there. So spontaneous generation of microorganisms does not exist. All living things arise from other living things, which is the cell theory. The cell theory in biology says that cells exist from pre-existing cells. Now, this is Louis Pasteur, which is the guy that we know these days on the issue of pasteurization, and that is uh, traceable to his name. Now, so this is what he did. So he also wants to confirm if microorganisms actually develop or come up spontaneously. And then he used this flask with curved necks. Now, so what he did is this. He realized that when the necks of the flask were tilted just the way they were tilted, so it saw that there was no um, microbial growth, rather. So he performed the experiment with a swath neck, and when the neck remained upright, there was no microbial growth that appeared. When the flask was tilted, dust from the bend in the neck switched back into the flask and made the fusion cloudy. Okay, let's go. I'll talk about that. So, um, all right, I think I'll talk about the pasteurization still later when I'm talking about fermentation. So this scientific method talks generally about that you have observation, you have questions. After you ask a question, you have hypothesis, and after hypothesis, you have a prediction. After prediction, you do an experiment or your prediction. After prediction, you do experiment. After experiment, you come up with results. Then your results could formulate a theory. So let me ask you, like I said, today is just a calm introduction, not teaching yet. What is the difference between a guess and hypothesis? What's different between guess and hypothesis? Guess, G, hypothesis is testable. Okay, thank you. That's number one. Hypothesis is testable. Guess is not testable. So I can just say that I feel like this is correct. Now, science doesn't have respect for my feelings. And so when my student answer question and say, I think the answer is this, I'm going to ask, no, tell me the answer. Because it's not feeling now. It's not what I feel like it is. So hypothesis must be testable. It must be falsifiable. It's not something I would just say, I think this is it. No, it's something that must be testable. Okay, any other response? What is the difference between guess and hypothesis? I'm waiting. Guess and hypothesis. I've only gotten one response, right? Hypothesis is testable. It can be falsifiable. Any other response? Logical reason for something happening. All right, thank you. It's logical reasoning. Now, when we say logical reasoning, it means reasoning that has to do with logic. And logical reasoning means A equals B, A plus B equals C. And I can explain 1 plus 2 equals 3. I can explain it by breaking it down, and it's still going to give me 3. Now, so that's why there's always a science behind hypothesis. There's something I talk about when I talk about hypothesis. So my 6-year-old, now when she was 3 years, she came one day and said, Daddy, my toy is not working. She had a piano and the piano stopped working. So I'm like, oh, it's the battery. So I went ahead and changed the battery. Now guess what? When every other toy stopped working, what do you think she's going to come back to tell me? She just come back and say, guess is prediction, hypothesis is observation. Okay, I'll connect that together. That's thoughtful. Now, every time every time any toy is not working, she's going to come and say, Daddy, the battery is bad. So it took me a long time to let her know that some toys are bad. Battery will not revive them. Okay, good. So the reason she's coming that I should change the battery is because I have changed the battery for a toy one time and that changed the story. The toy started working. So because of that knowledge, she's coming back and say it is the battery. It was a serious matter because sometimes she will be crying and say it's the battery. I'm like, no, we've changed the battery. The battery is not working. Now, but because she had a prior knowledge that battery could affect toys from working. So every research, every experiment, every hypothesis is based on prior knowledge, science knowledge. So in the lab, when we do any experiment, there's a science behind every experiment. You don't just go into the lab and mix things together and see if it will work. No, there is a science behind it. You should be able to explain the science. And that's why you notice that if you are going to have a research, either at the master's level or you want to have a research, you want to get a grant, you have to present the science of your research first. And then they look at it and say, okay, this is looking like something that can be spent on, that you can commit funds into. And then they work on it. Because nobody is going to fund a research that is just, I'm thinking this will work. No, it's that there are sciences you've put together, you've studied and have scientific background, literature review, and then that's what informed the hypothesis. Now, hypothesis is coming from an so sometimes just to make it a little light, we call it educated guess. And what we mean by educated guess is because it's coming from a knowledge, from something that you know, all right, but it's not guess actually. Now, all right, so thank you for your responses that I'm seeing on the chart, but that's hypothesis. Hypothesis coming that is coming from, of course, you could make a prediction from your hypothesis. Now, prediction is this. If you say, if your battery is, if your battery is not working now, I mean, if your phone is not working, you can say, okay, if I charge it, it's going to come. The hypothesis for that is that the battery is down. Now, prediction can now be, if I charge this phone, it will work. You are making that prediction from your hypothesis. So that's different from the random prediction because it's coming from your hypothesis. All right. Then you can make experiment based on the predictions you propose from the hypothesis if you have an hypothesis. All right. So looking at this chart, this is the framework of every research. Observation is the first thing. I was telling people that when people started dying because of COVID, if nobody paid attention and they just thought people were just dying, people will continue to die. Nobody will do anything. The same way that our car gives us signals that something is wrong, you just know that your car is not doing the way it used to do, but you didn't pay attention to it. You didn't do anything about it until it now stopped working. That's the same thing that could have happened when researchers or when scientists don't pay attention and not observe stuff. So observation is the beginning of every research. Observation. Second is to ask a question. Why is this thing like this? The third is then hypothesis. Now, after that, you can either have a prediction in between the hypothesis and then experiment your hypothesis. If your hypothesis work, great. You get your experimental data, your control groups, you get that. If the hypothesis work, you accept the hypothesis and then you formulate a theory. If your experiment does not work, then you reject the hypothesis or possibly modify the hypothesis and then you do the experiment again based on the modified hypothesis. All right, so that's the process of scientific research. All right, this just talk about that thing with, the scientific method. You can watch it on your own anytime. Now, the other thing they had challenge with that they had to figure out is the issue of fermentation. So in the bid to ferment wine, the wine got spoiled. So think about it that way. In the bid to ferment wine, the wine then again got spoiled. The issue is now about how fermentation can happen without spoilage. So spoiled wine threatened the livelihood of many grape growers, and then the wine producer funded research on how to produce alcohol without spoiling it. All right. How do we ferment alcohol without the alcohol spoiling because of microbial activity? All right. So the debate over the cause of fermentation reaction was also linked to the debate over spontaneous generation. So that made it huge. Okay. So this is the Pasteur experiment that I said I was going to talk about later. So some scientists believe that air was what caused fermentation. Some other people said it was living organism that caused fermentation. Some people just believe that if there's air, fermentation will happen. Some believe that microorganisms are involved in fermentation. So that was actually the problem that Pasteur solved. And that's where the issue of pasteurization, all of that comes into place. So this is the experiment he did. Just observe this with me. Look at it with me. This is what he worked on. So let me have my annotation on here. Now, so here he did the experiment. The key thing he wanted to do, so there's fermenting grapes here, and then he did the microscopic analysis of that, and it shows that there's yeast there, and there's also bacteria there. So the issue is now to know what is actually causing the fermentation. Is it air coming into the grape? Is it the yeast, or is it the bacteria? What is causing the fermentation? Now, number one, the first thing he did, he made an hypothesis that spontaneous fermentation just occur, as though fermentation occur without anything. It's just spontaneous. So what he did is this. He had the flask, and then so flasks of grape juice are heated up sufficiently so that the microorganisms are killed. Then on the second day, he closed it. So it is closed after heating it up. They closed it. So closing it means that, number one, he has killed all the bacteria, all the microorganisms that are there by heating it up. Then they covered it, so that means air will not be able to come in. So at the end of the day, fermentation did not happen in that flask. The juice remain free of microorganism as well. So there's no microorganism, there's no fermentation. So that proves that fermentation does not just happen spontaneously. It means that something will need to cause fermentation, because now we don't have air, we don't have yeast, and we don't have bacteria, and fermentation didn't happen. Number two, then, he made an hypothesis and say air is what ferments grape juice. So he used the flask with the neck, with the curved neck, so that at least air will be exposed to it. It will be exposed to air. So the air came into the flask. There's still no fermentation, and then there's no microorganism. So that means air does not cause fermentation. Then the third hypothesis he made, he said bacteria is the one that causes fermentation to happen and will cause the grape juice to become alcohol. He did that, and he also closed the flask. He heated it up and closed it. He realized that the bacteria reproduce. All right, so he realized that the bacteria reproduce, and then he noticed that there was acid. So what does that mean? He then concluded on that okay, bacteria does not cause fermentation because alcohol was not produced. He then concluded that um, bacteria will cause um, grape juice to be turned into an acid, not alcohol. So he changed the hypothesis. Then lastly, the last hypothesis that yeast is the one that ferments grape juice into alcohol. So he had a juice in a flask in an inoculated with yeast instead of bacteria in this case now. So he put yeast in it and covered it. So no hair, he covered it. And at the end of the day, he saw that yeast reproduced, more yeast was made, and alcohol was also produced. So he accepted that this hypothesis is correct. And so that was the hallmark of that experiment for Pasteur. He discovered that yeast is what is responsible for fermentation. All right, so let me see a few things I still want to touch base before we go. Now, I've talked about all of these here. Then these are some some industrial uses of microbes. You can read that up. Those are things that microorganisms are used for. So we've talked about that. So he developed the germ theory of disease. Some diseases are caused by specific germs called pathogens. All right. Now, the coach majorly is major research, or the major thing he came up with is the etiology of microorganisms, that they are pathogenic organisms. They cause diseases. So he demonstrated that bacterium, for instance, caused anthrax, and then he observed some of their colonies. So, I don't think I really need to talk about this research. We can go from, let me see if there's anything else I still need to touch before time goes off. All right, so these are different diseases, scientists that worked on them, the year they work on them, and the agents that causes the disease that they researched or that they highlighted. All right, this is, when you see this now, these are gram-negative bacteria. Some of you will see this very well in the lab. Later, you have to stain bacteria and look at which one is gram-positive, which one is gram-negative. So these are gram-negative and these are gram-positive. For gram-negative, they have enough peptidoglycan. I was telling you earlier about it. Peptidoglycan is what determines which one is gram-positive or which one is gram-negative. Gram-negative have less than 10% peptidoglycan in their cell wall, whereas gram-positive have about 90% peptidoglycan in their cell wall. So gram-positive have thicker cell membrane, thicker cell wall. Gram-negative have thinner cell wall. So the major thing that differentiates them is their peptidoglycan. The peptidoglycan is quite enough, about 90% in gram-positive, and that makes it to stain purple. And that's why we call it gram-positive. So the stain, Graham stain, the stain is called gram stain. The gram stain peaks on peptidoglycan and makes it turn purple. But the gram stain does not peak on the peptidoglycan on the gram-negative. It's not that peptidoglycan is not there. The only reason is that peptidoglycan there is very small. It's as though it's too small to be detected. That is the idea of gram-negative bacteria. All right. Um, Nightingale nursing, washing of hands, all of that. Yeah, let me see if there's anything else. So the issue of vaccine came into place, no infection control, chemotherapy. Those are the things that came up in the golden age. Um, all right. So this table here highlights the major scientists and the things that they did in between the process to initiate the golden age of microbiology that we study now. So all of these things, those are the like the Gram stain now. The guy that did it, his name is Graham, and that's why it's called Gram stain, right? Okay, so let me see if there's still anything. This talks about the areas of biology, of microbiology, and what they do in those areas, their description. All right, I don't see if there's anything else I still want to point out. All right, that's just okay. So that's it. So that's chapter one. Um, Best thing to do is to please watch. I mean, do the practice questions I posted so that you have a good view of